a pleasure to have here with us Professor Shai Revzan from the University of Michigan. Shai has an interesting and interdisciplinary set of research interests. His PhD is really in biomechanics, but he also has a master's degree in computer science. His doctoral work was at Berkeley. He actually does write patents as well, so he has several of those. He has worked in the industrial context quite a bit. He started a company as well. So, as I said, quite an interdisciplinary set of interests. Within those sets of interests, there is a very strong dynamical systems streak in the geometrical, proud geometrical tradition of Guggenheimer models and so on and so forth. The applications, let's say today, but who knows what's going to happen tomorrow, are within the context of bio-inspired locomotion. His lab at Michigan has a nice acronym, BIRDS. And that's, I guess, also related to the position of ecology and evolutionary biology. In Michigan, today he's actually talked to us, going to talk to us about models and what kind of challenges the models for those kinds of devices and those kinds of problems represents.
Thank you. So, and thank you for inviting me, and Igor in particular. So the content of this talk is also motivated very much by my collaboration with Igor. So rather than talking about all the great stuff I'm doing, I'm basically going to tell you how I failed for a decade trying to do something that maybe Igor can now help me do better. So it's going to be a talk that has a lot of old stuff. Some of it I've never actually shown in talks because it's the story of the failures, not of the successes. And so I'll start first saying, if I'm talking about 10 years of history, there are a lot of people who contributed. I couldn't even put all the faces on the slides. That list up there are most of the senior people who were involved, but there are a whole, and these are my current students who are involved, and a lot of universities and a lot of funding. But I can't really thank all of them enough for what has been a very, very interesting ride. So what am I going to talk about today? I'm going to talk about basically 3 parts. I'm going to talk a little bit about why we want to do data-driven modeling of legged locomotion. I'm then going to talk a lot about what happens when we try and look at legged locomotion as a limit cycle oscillator. And I'll shamelessly plug the book chapter I wrote with one of my students in this recent book on bio-inspired legged locomotion. That's chapter 3.5, and a paper we have that covers these things in a briefer form, if this is interesting to anyone. And then, because this is such a dismal tale of failure, I will end on a positive note with a few of the things that have been working for us lately. So all of you must be aware animals use legs. They use legs from animals as small as this flea to animals that are R-scale, so over a huge range of scales, multiple terrain types, and those are robust to many, many forms of failure. And here's what I mean by robustness. So here's a video of a cockroach running on a rough terrain. And here's a video of the same cockroach running on the rough terrain. Can anyone tell me what's the difference between the 2 cockroach videos? So it's not missing a leg, but you're close. In this video, the cockroach is missing all of its feet, not just one, all of them. All 6 feet are missing. The point is, there's no change in performance that we can measure. It basically looks identical. Try doing that with your car. Rip all the tires off, see how it works. Right? This is not even remotely close to the kind of robustness we achieve with our engineered systems. So the question is, how can we learn from biology to get this spectacular level of structural robustness and perform tasks reliably? And part of it is that control algorithm in animals is actually implemented in part in the mechanical structure and the material property and the emergent dynamics through the interaction. So when we look at this car suspension, we know that this is a shock absorber and there's a power transmission joint here. This is an ostrich suspension. Which part of the ostrich suspension does what? Right? It's not at all obvious. And that's part of what's so hard about functional morphology and about understanding what's happening in biology. And because I'm speaking to an engineering crowd, I had to put in this slide, which is a slight aside from my talk, and that is there's this huge pitfall when engineers go to learn from nature and do biomimicry, and that is any biologist will tell you screaming up and down, evolution doesn't optimize. Well, evolution doesn't optimize performance. It optimizes something called Darwinian fitness, which is the average number of viable offspring. That's what it optimizes, which means that it doesn't really depend on the thing you care about directly when you're looking at the performance of that organism, and most likely. It improves very little with being excellent at something, and often what it is sort of a tinkering and co-opting of existing features that the ancestors of that animal had. And the other thing is there are a lot of extraneous constraints, and anyone who does optimization realizes that constraints matter a lot. There are a lot of extraneous constraints on biological optimization. Animals grow. Imagine you had to design a car that works from this size to a size that seats 8 people. That would probably require a few design concessions for that car. Most arthropods have that sort of change in size over their growth. There's linkage, which is something people don't usually think about, but the way genes are laid out on chromosomes matter as to what evolves easily and what doesn't evolve easily. 2 genes that are on the same chromosome next to each other are almost always transmitted together to offspring. 2 genes that are on entirely separate chromosomes mix very quickly. So the direction evolution takes might have nothing to do with the function that you're looking at. It might have to do with the genomic layout of the organism that you're looking at. And of course, there's something that even Darwin knew when he first talked about evolution, and that's the notion of sexual selection. You know antlers? Antlers are a horrible idea. The only thing good about antlers is that the ladies find them sexy. That's the only thing that's good about antlers. And that's pretty obvious about antlers because they're big and difficult physiologically to create. But now imagine that you're talking about weakly electric fish. These are fish that swim in murky waters, and they use an electrical signal to see around them in the water and also to communicate with each other. And you wanted to build a sensor that's based on weakly electric fish. Is the song of a weakly electric fish that way because it's good for sensing, or is it just a sexy baritone? Right? You wouldn't know unless you did some pretty serious biological investigation. And so, because of all of these and many more considerations, whenever you think about learning from nature, please talk to a biologist who studies that organism. Because the answers, real answers that have value for engineering, require some careful biology. They're not just, hey, I looked at this, it looked cool, let's do what the animal did. Okay, so how do we study these complicated systems? Well, we usually study them using a hierarchy of models. And years ago, Full and Kodacek and Guckenheimer and Holmes together formulated these concepts that are called templates and anchors. It's basically the notion that you have a hierarchy of models. Anchored models are very closely tied to their morphology. This is an actual scan of an actual cockroach. Someone froze that cockroach, sliced it, scanned it, put it into an atom simulation. This is a very extreme abstraction where we say there's just a mass with a moment of inertia and one springy leg on a 2D plane. All of these are useful and informative, and the relationships between these models is informative. And I'm plugging another chapter that talks about templates and anchors if you want to understand what sort of notions make sense for templates and anchors. It's not just one thing. The kind of templates and anchors relationship that you'd want if you're doing modeling is different from what you want to do if you want to do synthesis of devices or other applications. So what we would love to do is find good template models that help us understand locomotion. We'd like to look at an animal and reduce it to these sort of essential models that help us understand what's going on. Okay, this is at least where I started as a wee grad student. And so one way to look at it is notice the fact that when an animal is running around, it's doing a repeating behavior. A repeating behavior that if you perturb, you come back from the perturbation to the repeating behavior is basically what a stable oscillator is. This is the definition of a stable oscillator. So there are things we know just by virtue of that. Smooth limit cycle oscillators always have a canonical phase coordinate. That phase coordinate partitions the state space into surfaces that are called isochrones. And basically, the phase information is the only information that persists over time. Everything else dies away. But if you perturbed in phase, that perturbation in phase will persist for a long time. And so this stuff has been known for a long time. So the first thing I set out to do is, okay, from 1,000 miles away looking at the running cockroaches that I tried to model, they're an oscillator. How do we pull out the phase? Well, if we manage to get that phase out, of course, there's a relationship, which is, as a function of phase, there's a characteristic motion of the center of mass, there's a characteristic motion of the limbs, and that can already be a really powerful idea, because we can use that to extrapolate what the animal would have done if we didn't perturb it, then look at a perturbation and see what we can understand about control by that discrepancy between the prediction and the animals. So the goal is to find a way to map the kinematics that are easy to measure. We look at the animal and we can measure points moving, map that to phase, map it back to a model of kinematics that we can then use. So the first thing I did was collect a lot of kinematics. In this case, is foot motion with high-speed video. This looks very easy. It's 3 years of my life as a grad student to come up with a good method. And then after, so this is the same motions you see here in 2D in the body frame are here over time. And then we can take these motions over time, plot the phase space of each leg, combine the phase spaces of the legs to some combined set of variables. And if you look at how this combined variable cycles around the origin, you get this nice gray trend line, which is, in a sense, a global phase for the animal. And if you look at the phases of individual legs, you can now look at the difference between these two lines, and you get this. And this is a pretty informative picture of how individual legs are changing their timing. The animal here is running in what's called a tripod gait. Tripod gait means these three legs in warm colors are moving together in sync, and the cold color legs are moving together in sync, and you see that here pretty clearly. There's a pi radian difference in phase between them. And you can also do a perturbation and see how phase changes. And so that's the next place I went to, is running an animal. So this is a camera tracking the animal, and we're doing a frame of reference for the camera that's tracking the body frame of the animal, tracking the legs. The animal is hitting an obstacle, and you see how the green model of phase before the impact changes. There's a phase shift, and then the frequency stabilizes again. So here there's a pretty big frequency, which is the slope of phase, and the frequency stabilizes again, but at a phase offset. And you think, oh, okay, why is that useful? Well, it turns out that if you take these three hypotheses of the control structure of an animal, and you say, is the animal just running feed forward, and it's just the body that's interacting mechanically with the environment? Or maybe the animal is dictating a pattern that's being followed by something like a PID controller, a tracking controller, and that's the dominant form of control. Or maybe the sensory information is modulating the pattern that the animal is trying to execute. These three hypotheses that seem very high-level neuroscience hypotheses can be separated by phase response experiments. So you can actually answer really interesting questions about the control in organisms just from these very external views, like phase response. So here's one of the things we did with that. Ran a cockroach on a plate that's getting yanked sideways. And just to give you a sense, the delta V induced sideways is twice the forward velocity of the cockroach. So this is like me being tackled by a sumo wrestler. But for cockroaches, that's not a big deal. You see the animal just rights itself and keeps on running. And so we look and we average this, and we look at the ensemble of animals, and we get a whole god-awful mess, right? So this is the perturbation here. You see the acceleration. There are error bars on that. It's pretty reliable perturbation. And the phase just drifts apart, and we have these really ugly lobes of distribution in various outcomes. This is this is forward velocity, this is lateral velocity, etc. So this looks like a complete mess. But one thing should always get you twitchy if you're looking at experimental data. If variability stays small and then inflates and then shrinks again, right? Variability normally, if you have no memory in a system, then the system variability increases and never decreases again. So if you see variability decreases, something's on here. And sure enough, if we use phase carefully, we actually realize that it matters which leg the animals were on when they were perturbed. If they were standing on the right tripod or left tripod, explains everything you see here. And it turns out that there are these two populations that you can separate out, and they respond differently. But the other really interesting thing, which was the motivation for this experiment and the paper that came out, is it turns out that although the animals could respond within about 20 milliseconds to the perturbation, they don't. They respond in about 150 milliseconds. So they keep moving willy-nilly like a clockwork toy for 3 steps after they were perturbed, because it turns out they seem to rely on the mechanics to handle the short-term recovery. And the nervous system only starts changing the motions at a substantial delay. And we couldn't have seen any of that if it weren't for treating these animals as oscillators, getting a really good, highly resolved hypothesis of what they would do unperturbed, so that we could tell the difference. So what I hope you get from this is this idea of recovering phase is actually a pretty powerful one. It's pretty useful. In fact, it was so useful that we then went ahead and developed an algorithm for recovering phase from data. And that algorithm is, I think, still pretty much the state of the art to this day, unfortunately. In terms of, if you have a bunch of time series from a nonlinear oscillator, you want to recover phase of that oscillator, you can run it through our algorithm, and it will give you phase. And just to impress upon you how much more powerful that is than previous methods, this is the statistics of phase error with several other methods that were used in the literature before. So that's the blue and the red, and our algorithm has these nice parabolas. So basically Gaussian error with very small variance in its recovery of phase. So, okay, check. Step 1 of modeling an animal as an oscillator. We know how to recover phase. In fact, then we can do something like convert an entire leg into an oscillator. Right? We take, this is data from a horse leg. We take all these markers in 3D, we run them through our algorithm, and we get this very, very boring thing that rotates at constant velocity, because that's what you expect an oscillator to do. So this is much easier to analyze and understand than that. And if you use this phase to model variables as opposed to more conventional things that people have used, for example, time since heel strike for human running. So if you try and look at the variability of, in this case it's a position in human running, blue is the variability you get in your model fit if you use heel strike timing. Red is what you get from our model. You see how substantially smaller the variability is, how much better the fit is when you use a phase that's derived from actually thinking about things in dynamical systems terms. Okay, so that's the power of that. But we can actually go a step further than that. We can take that horse and turn it into 4 oscillators. So now you see the horse walking around has 4 oscillators that are in fairly large phase angles to each other. And then the horse is going to keep walking for a bit. And at some point, he decides, well, no, I want to switch. I'm going to start trotting. And the phase relationship changes. So we can talk about different gaits instead of in the classical way that people have talked about what are the footfall timings. We can talk about gaits as expressions of phase differences between leg oscillators. And so we did that and looked at dog gaits. So you have a quadruped. There are 3 phase differences. It's a cube. You can have a dog run around and look step by step, where does it fall in the gait cube? And where it falls in the gait cube can change when it's walking on flat terrain and rough terrain. And we found some interesting things. For example, that walking spreads out towards trotting preferentially when the terrain becomes rough. That is, you could think that if the terrain is rough, it's random with respect to the animal, the phase differences would get randomized. But no, the animal, for some reason, is only expressing phase differences that are closer to the axis connecting walking to trotting than other directions. And we can use that to look at gait changes, and we can use that to study how symmetric is control in quadrupeds and these sorts of questions. So not only is phase useful for modeling whole animals or single limbs, we can also use coupled oscillator models to understand control or pull out features of control. And for example, this would tell me if I'm building a quadruped robot and when it's switching from walking on flat terrain to rough terrain, if I don't see this, I might be suspicious that it's doing something worse than it could be doing, because animals are pretty good at walking on rough terrain. There may be a reason why they're doing that. I'm being very cautious because this is the only study so far looking at this. So as a biologist, I can't say. Maybe this is a quirk of dogs. We didn't look at other organisms. We don't understand mechanistically why this is, but it's certainly a puzzling feature. Okay, so now we've had some successes. We may want to go to the next step. And unfortunately, this is where things start going awry. So there's this guy by the name of Floquet who a while back, a few years ago, published a paper on linear time periodic systems where he pointed out that solutions to linear time periodic systems split into two parts, a part that's cycle to cycle and a part that is within a cycle and periodic. And so why is that relevant to oscillators? Well, oscillators, if you look at the perturbation off an oscillator trajectory, then that's a linear time periodic system. So the first order approximation close to an oscillator's trajectory, the perturbation equation around that trajectory is going to be a linear time periodic system. And so you get the following sort of description for how perturbations behave. If I have a perturbation at phase theta, it's going to be related to the perturbation at phase phi by some periodic change of coordinates called here f. So we're doing f inverse at theta, and then we're going to convert what we get back to phase phi. But what we have in between here is just a diagonal linear system. It's the most boring LTI system imaginable. So this tells us something wonderful. This got me really excited when I learned this, because it said, if we find this change of coordinate, we just need to pick this f out. If we found this f, suddenly the system that looks really complicated and very hard to understand will just be an LTI system, which undergrad controls classes tell us how to control. We know how to understand its invariant subspaces. We understand everything that's going on there. And here's another thing that's really exciting about that. This idea of templates and anchors really has to do with relaxation times, right? If I have a system that has a low dimensional manifold that it's occupying most of the time, and this is this blue space, perturbations off of this manifold in red are going to decay quickly. Perturbations within this manifold will decay slowly. So I'll be able to identify these template models in a data-driven way, whereas all the previous templates that were on that slide before, and since then a dozen more templates have been published in the literature, they're all created by someone being brilliant. This is not brilliance. This is number crunching. This would be great. And just to give you a sense of what sort of power this gives, so I made a toy model. This is a two-legged bunny. It just has two legs. They're coupled to each other unidirectionally. And if I perturb them at different times, so I'm doing this exact same perturbation on the exact same leg at different times in the cycle, you get very different responses. So if you're doing linear system ID and you see three responses like this, you go, oops, I can't do anything with that. This is obviously not a linear system. But what I can do is I can take that Floquet structure, and I actually know that this system has three modes. One mode decays very quickly. So it's a four-dimensional system. It has a limit cycle direction and three additional directions in its state space. There's one mode that decays quickly, two modes that decay less quickly. These two modes that decay less quickly, after you correct for their decay rate, give you these periodic coordinates that are a function of the place on the cycle. You take those same perturbations and plot them in these coordinates, and they look like this. Right? They're just abysmally boring. They're just decaying exponentials. So each perturbation, the blue perturbation, is these two initial conditions. The magenta one, these two initial conditions. The yellow one, these two initial conditions of this extremely boring linear system. So this description is very simple compared to this description where we don't know what the heck's happening. Okay, so this is why this could be wonderfully exciting if we could do it.
How did you get the coordinates for those things to look so good?
How I got those coordinates, I took a Poincare section, I took the eigenvectors of the Poincare section, and I evolved them through the ODE integrator. Okay. Now the problem when we want to compute these is that when we want to compute them, we're going to have to estimate all these linear operators out of data. And if we want to estimate linear operators out of data, we're going to get variability. So I'll point out that as I go forward through this talk, every regression I'm talking about, I'm going to bootstrap. So that allows me, if I have data, to balance the quantity of data from different experiments and to give distributions of the parameters that I'm estimating rather than just individual results. I won't go into the other points here, but just to say, okay, so how do we do this data-driven Floquet analysis? We start with an ensemble of trajectories in state space. We fit a cycle through them by having that phase variable. Once I have the phase variable, I can average all the points that had the same phase, and I'm going to get this limit cycle behavior. I could then pick a section on that limit cycle, find its return map, take the eigenvectors of that return map, and evolve them forward in time. And everything should be fine. In fact, that's what I did on that example, as you just asked me. So see, there are a lot of steps here. Okay, there are a lot of steps. We need to get lots of data from that rhythmic behavior. We need to compute that typical cycle that comes from averaging. We need to choose a consistent section for all the data sets that we have. Estimate the first return map. Find those eigen directions, those independent perturbation direction, and extend them around the cycle to get Floquet modes. The problem is each of these has problems. When I set out to do this was still as a grad student, I discovered that every one of these steps, I needed to do something that was not in the textbooks. So there was a lot of customization involved in trying to get this solution working. And as soon as we first published any result with this, reviewers immediately said, but you don't have enough data from a single animal, how do we know that this means anything? And then yours truly had to go on this adventure, which is build a test setup that can collect hundreds of strides from a single cockroach. And to do that, I had a computer-controlled mirror tracking this cockroach that's running on a transparent arena. And there's another high-speed camera that's looking over the shoulder of this webcam. So you see the webcam has this wide-angle view, and then the high-speed camera has a zoom lens that's focused in. And you see these little fiduciary points on the arena? We can then use them to correct the optics so that we get now a rectified video of an animal, and now we can track the legs. So you can imagine this was another year of my life. Okay, this is another year of my life just to get one additional data set to satisfy reviewers. And here's the sad result. So if we take the eigenvalue distribution that I get, so here the matrix of data I'm talking about is 27 dimensional. I'm doing bootstrap regression, so I'm doing multiple regressions of these 27 dimensional matrices, taking their eigenvalues, plotting the density of these eigenvalues on the complex plane. Two things should be apparent. Multiple animals and single animal, they look kind of alike except for this big round blob in the middle. So the lobe that sticks out here is actually pretty similar. But there's also another thing here which is really dissatisfying, and that is that these 9 slices here were taken in different places in the cycle, and these 9 slices were taken in different places in the cycle. And any of you who've read a book on nonlinear oscillators might know that the Poincare sections in different places on the cycle are conjugate, their linearizations are conjugate to each other, which means they are supposed to have the same spectrum. So I'm supposed to get the same picture at every slice. And these pictures are not the same. In fact, after you do the bootstraps, the blobs are somewhat similar, but if you just did individual attempts, they would be all over the place, completely dissimilar. That was really, really discouraging. So there was no way to get a consistent picture of what the eigenvalues are, which are supposed to be consistent everywhere along the cycle. And also, sort of, what do we do with this blob that appears there? So it turns out the blob actually comes from something that we understood later. Someone who knows the theory of random matrices may recognize something called the circular law. So it turns out that if you have a Gaussian random matrix, you look at its spectrum, that spectrum is distributed on a disk. If that matrix is infinite dimensional, that disk is just uniform, but for finite dimensional disks, you have this squishing onto the real line that happens as well. And I'll also say a bit about what you see here. So here we're putting a null model, which is, let's assume that the model of what our animal is doing is just going to be a random matrix. It's going to fail miserably. And so when we try and see what's the variance of the residual of our model compared to the original variance of each variable, these are different state variables in our system, you see that all of these are larger than 1, which is what you expect. If you have a terrible model, it's going to be worse than no model at all. Okay? And if we look at what happens after one step in the real data, you see so this is not a full stride, this is a step. So this is half a cycle, I should point out, which is why that's different from what you saw in the previous plot. There are clumps here. You can believe that there are collections of eigenvalues there that might be somewhat consistent. Some of the variables are being predicted. Those little bars here are less than 1. And that seems to be about what you can get with real animal data. In fact, even the real animal called human has a very similar eigenvalue distribution. This is from a paper I published in 2014 with some colleagues where we produced what I think is the only predictive model. Maybe actually last year something came out that is also somewhat predictive, but pretty much the only predictive model of human running behavior. So if you ask, what do humans do step to step to stabilize running? We could apply these methods to get some insights as to what's happening in human control, and postulate a model and postulate what's missing in existing models that attempt to understand what's happening in human running. And
Taking out the eigenvalues that are in the circle? Because you only have the ones that dissipate.
So I'm completely, these are Poincare section eigenvalues. They don't contain the unitary eigenvalue that's associated with the limit cycle. Yeah. These are cross section. Yeah.
When you collected data, I guess you were trying to make all the experiments kept so that there would be disturbances.
Yeah, this is free running animals or humans free running on a treadmill. I mean, treadmills exert some constraints, but we've actually controlled for that to check whether there's a difference in the control that you see close when someone's close to the edge of the treadmill or stuff like that. It wasn't a large effect.
And as they get tired, does it begin to change?
So humans do something really annoying, which is about on the order of 4 minutes, humans change how they run. There's a long-term trend. So these are 20-minute runs. So we ended up having to subtract a 32 stride baseline average before we could get anything meaningful, because that slow drift has an amplitude that dominates everything else and just kills the analysis if you don't subtract it out.
And the cockroach doesn't get
The cockroaches, so cockroaches really love running. One of the reasons we worked on them is, in fact, cockroaches are so geared to running that if you cut their brain off, they just run until they die of starvation. Okay, their default is running. They have these little hairs on the back that detect toads or other predators approaching them. They detect airflow, and it's been proven that the legs start running before the signal reaches the brain when that happens. So they are the best model for running in this regard, right? They love running. So, however, they also switch behaviors slightly on the order of about 20 steps. Okay, so what's the problem here? Well, the problem is that the spectrum, so this is, by the way, the projection of the density of the spectrum on the x-axis, where we've sorted eigenvalues by the real part and collected them in little bell curves. The problem is that these eigenvalues are very close together compared to the uncertainty in them. And when eigenvalues are close together, which eigenvector is associated with which becomes blurred out. You basically get a subspace rather than, you get invariant subspace rather than invariant vector. Or instead of a one-dimensional subspace, a higher dimensional subspace. So maybe isolated clusters of eigenvalues have stable subspaces, but can we get at those? So a way to think about that is this object called the Grassmann manifold. The Grassmann manifold is the space of all subspaces of dimension p in a linear space of dimension n. So for example, GR31 are lines in 3D. So they're equivalent to a sphere with antipodal points identified, or the projective 2 sphere, projective 2 space. But so the question now becomes, can we do an average on the Grassmannian? That's weird. Doing averages on curved manifolds is hard, but it turns out that you can try and do that by just doing a naive gradient search, because there is a metric on the Grassmannian. You can measure distance. So you can say, okay, let me just try and optimize what is the point that has the least average distance in terms of the Grassmannian metrics from all the points that I have. And that might be a good consensus subspace that we could use. Unfortunately, the Grassmannian is like a sphere. It doesn't have a global chart. You need to switch charts. There are a bunch of papers to read and to do that. And so we got a centroid and a radius that we might be able to get using this. But now we were asking ourselves, well, do these mean anything? Like, what would happen due to noise alone? And so how do we ask the statistical question? Well, that statistical question is really talking about probabilities, which are a measure of volume. They're a measure on the space. So one way to do that is think about what's volume of the Grassmannian as a function of radius. It turns out that this is known. It's a hypergeometric function that's really hard to calculate. But instead of using that hypergeometric function to decide what our threshold values are for something statistically significant versus due to noise, right? Because if we're spread out very far, we're probably due to noise. If we're concentrated more than you'd expect by chance alone, then it's probably significant. Instead of computing that hypergeometric code, we can just do that by generating uniformly random subspaces and tallying how many of them there are at any given distance, and get an approximation to these distributions, and then get the following lovely video which says, if I include more and more eigenvectors in my subspace, this is the distribution I get. This is the null distribution that I have from random matrix theory, and here they crossed. So there was this threshold at which we suddenly could no longer distinguish. We added too many eigenvalues. When we had the eigenvalues that are close to the origin, those started blurring everything out, and it was like noise alone. So we went ahead and we tried to build that into a better model. So we have a paper from 2012 where we use this sort of coupled van der Pol oscillator system to make a system where we can design in the eigenvalues that we want to test this. And then we look at, these are the true eigenvalues we know because we designed the system in. So the ordinal of the eigenvalue versus its magnitude, just to have something that we can look at. And the green strips you see here are what you would get from a random matrix model of that dimension. So something that fits within the thick green line here is indistinguishable from the eigenvalue distribution you'd get from noise. And what you see with the blue, red, and green is that we amped up the noise in our system from a low level to a high level. Actually, the labels here are reversed. This is 0.9, and this is 0.09, and this is 0.01, and this one's correct. So as you see, when the noise is lower, we can identify more of the true eigenvalues. And if the noise is high, they get hidden in the noise. And so we can now do that on the real animal data. This is what we have in the real animal data. The gray line here is what you get if you do a random matrix model up to 21 dimensions. How did we pick 21? Well, what we did is we looked at how well this distribution matches the random matrix distribution in various sizes, and that match decreases. The error decreases more and more and more until we reach 21 dimensions, and then the error explodes up because it doesn't match anymore. And so it's pretty clear that this is the largest noise model that fits. Just to be sure, we compared with what are called surrogates, where you scramble the data so that it doesn't have some of the structure, but not other, and then that jump disappears. So it's pretty clear that this statistical test can let us know which part of our model that we got from regression is statistically significant and which part might be due to noise alone. And sadly, in my tale of dismal failure, this is as much as we could get. That is, I could never get those eigenspaces, those flow K modes in real data sets. The most I could get in the years I spent working on this was a statistical test to tell me which part is significant and which part isn't. But the part that I know is significant, I have no way of modeling other than knowing its spectrum. I've never managed to go beyond that. So hopefully everyone's sufficiently sad. Now I'll go to where things get a bit better. So in the last year or so, we've hit on a new class of simplified models that has me pretty excited, and they're based on ideas from geometric mechanics. So in sort of a very, very general sense, geometric mechanics come from the following observation. You describe your laws of physics with some Lagrangian, but that Lagrangian should be invariant under a group action. Which group? Well, where I do my physics on the table or where I do it in the world shouldn't change physics, right? It should be invariant under the group of rigid body motions, which means that if I change my positions and change my velocities in a compatible way, I should get the same value of the Lagrangian. So this is not a new idea. In fact, a bunch of people worked this out quite a few years ago, about 30 years ago. Bloch and Marsden and Murray and Krishna Prasad wrote this lovely paper where we can it asks the following, or allows us to answer the following question. As we cycle through our shape space of the animal or the robot, where do we move on this group of rigid body motions? So the vertical direction here is SE3 or SE2 is the relevant group of rigid body motions. And as we change shape, we also move. If we're doing a periodic motion, we can do cycles here. At the end of a cycle, we're going to end up having moved somewhere in the group. If that's a good motion, then we can repeat that and move effectively through space. And the key insight behind this that's very powerful is they showed that if the Lagrangian obeys this sort of symmetry, you can rewrite the equations of motion with these two parts. The top part here has the group variable, so this is the motion through space. It has a leading term which is geometric. So notice it just has our dot. It just has shape velocities, the rate with which I change my shape, feeding directly to the rate I'm moving through space. What's an example of that? If I'm doing this with my legs, the rate at which I'm changing my joint angles is exactly linearly related to the rate with which my body is moving through space. Right? Inertia doesn't play any role in that. Inertia comes in this p variable, which has its own dynamics. But this is not a restriction. This is a completely general theory. You can write any mechanical problem in these coordinates if you choose to. It's just going to be gnarly. Okay. But where things get interesting is what's called the principal kinematic case. What happens if you have so many constraints that you can rule out the existence of p? That is, if the dimension of that momentum in the equations goes to 0, then you just have a purely geometric system. The classical obvious example that no one thinks about is what I just did, for example, with both my legs on the ground, they're not only pin joints, they're not rotating, right? They're restricted in torque. How I place my legs entirely places my body, right? You don't need to know anything about momentum. If you know the angles in my joint, you know where my center of mass is, that's it. No momentum needed. But note that when we build robots, we usually control the ego shape, the shape of the robot, very tightly with PID loops. So that's something that is actually technologically achievable. This is what we usually do. So if we're in this world, and you look at this, this looks basically like a line integral. Right? There's something linearly multiplying the velocity of our trajectory. You could hope that if we're doing a loop on a line integral, Stokes' theorem can apply. And it turns out that kind of, sort of, I don't want to get into those details right now because the problem is that we're solving an ODE on a group that's not necessarily abelian and not necessarily flat. It has curvature in the group itself. So there are a bunch of caveats here. But the short version is this is approximately true and can be made much closer to being true if you pick your coordinate frame very carefully when you decide where you place this G in the body. And people have solved that. And so one of the things we did was ask, okay, if we have this A here, instead of estimating that Floquet structure, let's estimate A. That might be fun. That's a simpler object. And so we did that. We did a big regression for A. And that regression actually enabled us to optimize very high dimensional gate spaces. So we take, say, in the paper that I'm mentioning here, we take a 9-segment swimmer. Each of those 8 joints that it has is parameterized by a high-order Fourier series. So the overall model is 300 dimensional. If we wanted to compute the gradient of how it moves with respect to those inputs, that would be infeasible in terms of time. But instead what we do is we execute a behavior with some noise. We identify A around the limit cycle, essentially with the same approach that I described before, this kind of phase-based regression. And then we use the model that we just derived to compute the gradient with respect to all the control inputs, which is really fast because this is a very succinct model. And then we can do an optimization step, a gradient ascent step, rinse and repeat, and it works pretty well. It also works not too badly on actual hardware. So this is a uselessly moving robot, and after 2 iterations, this robot is crawling on the floor. So it's the same algorithm. It's just assuming that the system is geometric, that there's no momentum. Okay, so we decided, well, maybe this is a good idea. And now what makes this a good idea is, of course, momentum is lost through dissipation, through friction. And in fact, we already knew that when our robots move around, they slip a lot. All the highlighted dots here are legs slipping with respect to the ground. So there's a lot of slipping going on in the robots. There's a lot of slipping going on in cockroaches. The red here shows you how much they're slipping. So in fact, about 20% of the distance they travel in the world frame, they travel while sliding backwards on the ground, which is something we don't like as humans, right? Bipeds slip, we break something, right? We're big, we're tall, we rely on one leg to support us at a time. If that leg slips, it's a tragedy. But a cockroach can lift all its legs off the ground and just stay on its belly. I mean, nothing will happen. So they're obeying a different set of rules. So this led us to think, well, let's try and build a connection-like model. Let's try and now, instead of doing a data-driven model, let's do a theory-driven model, but we're going to shoehorn that model to be a connection. So we want the inputs to be leg motions. We want the outputs to be body motion in the world frame of reference. And we basically assume it's a plate that tilts at small angles in pitch and roll and has springy legs sticking down. And to make it behave like a connection, something needs to multiply velocity. And so we decided to do horrible and unspeakable things to Coulomb friction. And whereas Coulomb friction depends on v hat, only on the direction of your velocity sliding on the ground, we decided to put actual v there. That is, make it depend linearly on how fast you're sliding on the ground. This does something really weird because it basically says, a leg that's not sliding on the ground is not generating any force. Okay, this is the opposite of what non-slipping creatures like us like to believe. But then, doing that, we now assume that the entire system is in force balance and try to solve the equations. So the way we go about that solution is we postulate pitch and roll angles. We figure out how far we need to move that plate down so that enough legs are contacting the ground to offset gravity. Now that we know which legs are in contact, we can balance the moment in Z and the force equations. All of that behaves like a connection so far. All of this is entirely linear because these are homogeneous constraints. These are Pfaffian constraints, as people in mechanics sometimes call them. The problem is that we might still not meet these two equations, the moment x and moment y equations. So here we iterate changing alpha x and alpha y because I'm lazy. Because actually alpha x and alpha y, if you look at what the equations are that you need to solve, these are coupled quadratic equations. So they're 0 to 4 solutions. This is not a very tough equation to solve, but we just solved it iteratively with basically an F-min. And then we tried this on some data from our robots. So the blue line is what the robot's actually doing. The red line is what our model is predicting. The red line is noisier because what we've done is we've assumed force balance, which is basically saying everything has reached its critical velocity. Critical velocity is something you reach at time equal infinity. We've compressed time. When you compress time, you get high, moderate frequencies become high frequencies. So say, okay, this is really seems noisy. Let's just match the spectrum, the power spectrum of the robot data to see if we remove those high frequency things that are non-physical. And that's what the green line is. So if you look at both of these plots, the red is what came directly out of our simulation. The green is after you take the red and just filter it to match the power spectrum of the real motion. So it's not any deep modeling step. And this was disturbingly good, right? You don't expect a model that's so idiotic to match little wiggles in your theta dot and your x dot. So I said, okay, let's try it on another robot. Same thing, right? Works really, really well. So I decided, okay, let's get ambitious and do it on a robot where it shouldn't work. Because these are hexapods. They have 3 points of contact with the ground. So we're pretty sure they don't have momentum because there are no unactuated directions. Let's go with quadrupeds that have 2 legs in contact with the ground, and therefore they have one unactuated direction of pitching over the line between the 2 points in contact. So we contacted a guy that you guys, as it turns out, saw recently, Kaushik Jarayam, working at Harvard, lent us some of their hammer robot data. And this is what we get with hammer jumping. It's pretty good. With hammer bounding, so-so. With trotting, something weird is happening. We're not yet sure what's happening with trotting. But if you look at the other variables for trotting, well, y actually gets matched pretty well. Theta gets matched pretty well. But something horrible is happening to x. And to close off back to where I started, we try and do this to the cockroach. The model tells us something completely nuts. But if you filter it to the power spectrum of the actual cockroach motions, you get the green line, which is still pretty bad, but having looked at cockroach data for many years, this is about as good as anyone else's model. So maybe slightly more optimistic, this form of data-driven modeling for multi-legged locomotion. And thank you all for your time. This is all I've got.